Here. Hey, Austin. Macromolecules today. You got that handout? If not, it's on the screen. Can we see it? Big enough? Nope, that's cholesterol. The macromolecules will say macromolecules. Find it. Um, as the name suggests, big molecules. Big is relative. We just mean bigger than like benzene. Uh, um, the four classes that we're going to describe are carbohydrates, lipids, proteins, and nucleic acids. If we summarize the class with maybe important properties, what would you say a carbohydrate? Why would we study them? They're a food source, an energy source. Who makes them? Plants. And of course, are they carbon based? And most of your carbohydrates are derived from sugars. So not all carbohydrates are sugars, but all sugars are carbohydrates. And if we rattle off some names like glucose, fructose, lactose, maltose, those are all types of sugars. If we polymerize sugars, then we can get starches, cellulose, etc. So stepping back a second, if we say monosaccharide, that's another name for a simple sugar. Disaccharide would be two monosaccharides linked together, and polysaccharide would be many monosaccharides linked together. So, of course, since this is organic chemistry, are we interested in the structure and how they're linked together? We are. A lot of words. We're going to look at the picture. So if we look at that picture, do we see the molecule in the center that's labeled glucose aldehyde form? And would we all agree why it's an aldehyde? And when we draw a glucose like that, we're always putting the aldehyde on top. So is it this bonding pattern or that carbon, which is what we're referring to as the aldehyde? And then following, going down, is it 6 carbons in a chain? And do we see lots of OHs? And could we say that that glucose is a polyol, many alcohols? But is it also an aldehyde? Of particular importance is this carbon, which we were going to call carbon number 5. Its OH is projected to the right, and when we use this convention, we're using what's called a Fisher projection. And when that carbon, called the anomeric carbon, is projected to the right, that matches the original glucose molecule that was found in nature, and that was always D. So this is D-glucose, and we are referencing D-glyceraldehyde. So in sugar chemistry, we could use RNS, but the convention that most use are D and L. And it's this carbon that makes it D or L. So if this OH goes to the right, that's D, and that's the most common form of most of your sugars. Equally important about this carbon and that OH is that's the one that wraps around and is able to cyclize on the aldehyde to make these hemiacetal versions. And as you could see by those arrows, those are equilibrium arrows. So it's not resonance. These are actually moving atoms and electrons. So they are in equilibrium with each other. And you can debate a lot as to which one's more stable, which one's more common. But more importantly, it's important to know that when we say we have D-glucose, it can really be any of those 3 structures. And what's really important for what's coming up is to recognize that if the OH goes down relative to the H, that's called alpha. And if the OH goes up, that is called beta. And from our past cyclohexane, would we say that the beta version is more stable chemically because it has the OH in the equatorial position? That said, all bets are off when we start doing reactions of these, because most of the reactions that we're going to encounter in biology are all enzymatically catalyzed, so the enzyme can pick and choose what it wants to do for whatever reasons. So beware that are all three of those molecules showing glucose, and we would call this the aldehyde form, and these two are the hemiacetal form. And the hemiacetal again forms when this O on C5 wraps around and cyclizes on the aldehyde carbon. Similar to that, here's pictures for sugars we'll study when we make nucleic acids. So this is ribose, and this is deoxyribose. And of course, you're probably familiar with DNA and RNA, which is where you would find these sugars. So as a quick quiz question, which of those sugars is found in DNA? Deoxyribose. Which one's found in RNA? Ribose. And what's the difference between ribose and deoxyribose? Ribose has this OH, deoxyribose doesn't. So that's the only difference between ribose and deoxyribose. Other than that, are they the same in that they both have an aldehyde version? And do they also have this cyclic hemiacetal version? And if we are to justify D or L, we would look to this C furthest away from the aldehyde, that is chiral. And if the OH goes to the right, that's again D. So this C with the OH to the right is again D. And here's another sugar you should be familiar with. This one is not an aldehyde, but a ketone. And it's called fructose. And so analogous to glucose, 6 carbons total. C5 I've got labeled here. Quick quiz, if the C5OH goes to the right, is that D or L? D. And if this cyclizes, does it make a hemiketal or hemiacetal? Now it makes a hemiketal because the open chain form is a ketone. So now is when you'll realize you're not going to be asked to draw these on a test, but will you be asked to recognize them? So if we scroll back up, would you say that glucose is a 6-carbon aldehyde? And does it cyclize to a hemiacetal? And ribose and deoxyribose have how many total carbons? 5. So unlike glucose, are the riboses C5 aldehydes. And glucose was a C6 aldehyde. So somewhat naturally, when these cyclize, do they make 5-membered rings or 6-membered rings? 5-membered rings. So that's a way you can sort of identify them. And is it pretty easy to say deoxyribose is the one without the O right there? And would we call that carbon number? If we were going to number this, where would we start numbering? The aldehyde carbon, 2, 3, 4, 5. And so where is this aldehyde carbon in this cyclic version? Right there. So this is 2, 3, 4, 5. So common question would be, is this 2, 3, or 4 deoxyribose? 2, because this is carbon number 1, so this is carbon number 2. So what makes deoxyribose different than ribose is it's missing the 2OH. And then lastly, fructose is not an aldehyde at all. It's a ketone, and the ketone obviously has to be on carbon number 2. And that carbon number 2 in the cyclic version is right there. So is it again the C5OH that wraps around and cyclizes? So those are what we would use as examples of monosaccharides. So mono just means one. That'll be obvious when we go to this. So if we take this version of glucose, and quick quiz, if this is glucose, which form is shown? Open chain or hemiacetal? That's the hemiacetal, and what makes it a hemiacetal is again COCOH. So this isn't really just an alcohol anymore, and this isn't really just an ether, because you have this ether-like linkage connected to OH that's called a hemiacetal. And that's a key feature you want to pick up on. So over here, do we have COCOH? So that's now the hemiketal version of fructose. This is the hemiacetal version of glucose. And when we dehydrate them, what are we going to remove? We're going to remove OH and H. And when we do so from between two molecules, we would call that a condensation. And then do we get this new molecule, which we're going to call a disaccharide, because we now have two monosaccharides linked together. And this disaccharide's called glucose 1,2-fructose. And importantly, glucose alpha. And we're calling it alpha because the connection from one sugar to the next goes down. If this connection went up, then it would be beta. And that's going to have huge implications coming up. So chemists and biologists get tired of saying glucose alpha 1,2-fructose, and they make up a new name, and they say the new name we're going to call sucrose. Just to make sure you're making a connection to your real life, where would you get some sucrose from? If you have a kitchen with a big bag of sugar in it, that is sucrose. So would you say your big bag of sugar in your kitchen is a disaccharide? So we're setting up for one important part about carbohydrate chemistry, and that is hydrolysis. But before we get there, we're going to describe a different type of disaccharide. In this example, are we just linking two glucoses together? And in this example, is this hemiacetal version alpha or beta? Beta. And what makes it beta? The word or the fact that the OH goes up relative to the H? Up. So now when we dehydrate, now I'm not connecting from 1 to 2. Now I'm connecting from the carbon number 1 of the first glucose to carbon number 4 of the second. And the connection is called beta, again, because the OH off the first sugar is up. So when we connect this C1 to C4 through this linkage, do you get why it's called beta 1,4-diglucose? Or glucose beta 1,4-glucose. So how are we to recognize that there's two glucoses? Go back and look at the glucose structure until you're familiar seeing it. How do we know it's beta? Because this connection from the first sugar to the second one goes up. And how do we know that it's 1 and 4? Because when we look at these glucoses, this is carbon number 1, 2, 3, 4, 5, and 6. So can we make sense of that name? And if we do that a few more thousand times, would we still call it a disaccharide or a polysaccharide? A polysaccharide. So this right here would be called a disaccharide. Now, if this N is 1,000 or so repeating units, then it's called a polysaccharide. And the key thing about polysaccharides is the type of connection gives the properties. So if the connection were alpha for all of the connections, then you would be at a molecule like starch or dextrin. If the connection is beta, the polysaccharide is going to fall into the class of cellulose. And to make that context, I would just say, would you build a house out of a potato? Does a potato have a lot of starch? Would you eat wood? Could you eat wood? Yes. Could you effectively digest wood? So when we say cellulose, we're speaking of, well, the purest form of cellulose that I could think of would be cotton. But you can also say wood has got a lot of cellulose in it. So as compared to, say, pasta, potatoes, rice, they have a lot of starch. So which one for humans is more easily digested? Wood or starch? Starch. And what's the reason that we're able to digest starches? Is because in starch, the connection from one sugar to the next is always alpha. Why can't we digest wood or cellulose or cotton? Because the connection is beta. So you should think of your beta connection as more rigid. We don't have enzymes that are able to digest it, meaning break it up into smaller pieces? And so can we use wood as a structural thing? Can we use cotton as a fabric? And then on the other side of the spectrum, is a potato sugar, or is a potato largely starch? But can we break down the starch and liberate sugars? And so if when we make polysaccharides, we're dehydrating, what do you guess has to happen when you break down polysaccharides? Said another way, what's the opposite of dehydration? Hydration or hydrolysis. So what's the product of hydrolysis of starch? Many glucoses. Why can't we hydrolyze cotton or wood? Because the beta connection, we don't have the right enzyme profile to break that beta connection. How about a termite? A termite would. So can termites eat wood? Or dismay? Yes. So that's really the first class of molecules. To summarize, of course, can we just burn sugar for energy? Yes. Are some sugars polymers that we can burn for energy if we can hydrolyze them? Are other sugars structural? And just because it's up here on this handout, has everyone heard of glycogen? Where would you find some? If you're going to run a marathon, what do you hope you have a lot of stores of? In the form of glycogen, which would be found in your muscles and your liver. And what happens if you run out of glycogen during your marathon? You're going to run out of energy and hit the wall. That's not a fun feeling. So glycogen is kind of how we store energy within our muscles and our liver. And I think we have about 200 grams in our liver and probably about the same amount in muscles, enough to get us through half a marathon. So that's why if you're going for the full marathon, do you have to eat? Or you won't make it. And if you train, do you basically teach your body how to store more glycogen? That's one of the things about training. So that's carbohydrates. Next up is lipids. So lipids from our lecture material we've already covered. Are they all things that don't dissolve in water? So the word lipophilic means fat soluble. Within the class of lipids are there fats, oils, waxes, cholesterol, etc. And to go back and forth between lecture and lab, would we say benzene is more lipophilic or hydrophilic? How about hexane? Anything nonpolar is going to be lipophilic, meaning loves lipids. So the way you remember that is lipids are nonpolar, they don't dissolve in water. So anything that's nonpolar is going to be lipophilic. How about ethanol? Lipophilic or hydrophilic? So ethanol's got that OH group. It's a pretty short carbon chain. It's going to dissolve in water, hydrophilic. And the key lipids that we've already talked about are triacylglycerols. So that's when you take this glycerol molecule, which is a triol, and esterify it using a carboxylic acid. And this R group is usually 12 to 20 carbons long. And this is one esterified alcohol. And now if you do that 3 times, you have a triacylglycerol. And of course, if this R group is 12 to 20 carbons long, are those all nonpolar and lipophilic? And to review, those are all of the fatty acids that are common. And what can we say they all have? An acid molecule, but a long nonpolar hydrocarbon chain. So there's an example of the full version of a triacylglycerol. So if we were to say, why is this lipophilic? Because of this region or because of that region? That region. So a key concept now is, if you have 2 of these molecules, how will they orient to each other? They will orient themselves in what we call tail to tail. So if you take this molecule, flip it over, you have naturally drawn the 2 tails close to each other. And why would the 2 tails want to interact with each other? Because they're both lipophilic. And if we do that repeatedly, do you get why it's called a bilayer? And then if you take that bilayer, can you envision wrapping it around into a circular or spherical shape? And then would that bilayer have a very nonpolar internal region? And then would it have a somewhat polar ester group region on the outside? And where we're going with that thought is, are these bilayers of lipids the cell membranes? So if your cells didn't have a membrane, would everything in the cytoplasm and nucleus just leak out everywhere and we would not have life? So life needs compartmentalization. How do we compartmentalize stuff? Within a lipid bilayer. And so if we ask, is the internal of the bilayer polar or nonpolar? Nonpolar. So would we say we now need another mechanism to let things get in and out, or we have to facilitate transport? Because water's not going to be able to get into your cells through the nonpolar region unless somehow it gets facilitated. So we're not going to make this biochemistry. We're just trying to realize that biological molecules might not have 3 totally nonpolar carbon groups. Might they have this phosphorylated ester if it's a specific membrane for a specific cell? So if we ask the question, which should be obvious, are the membranes around brain cells different than around liver cells? So they're going to be tailor-made for the cells that they're made for. So you'll hear the word phospholipid when you get to biochemistry. It's just a normal lipid, but instead of an ester, you get this phosphorylated ester. So that's just going to alter the properties of the membrane so that the cell can exist. And then we're on to cartoon characters, which will make more sense after the next section. But if this is a polar head group, and this is a tail, and this is a tail and a polar head group, this right now represents a lipid bilayer that's going to be surrounding a cell. And this blob right there is a cartoon for a protein. So would we say that proteins can span the bilayer? And why might a protein be incorporated into the lipid membrane? Several reasons. Might it assist in getting stuff across? Those we might call gated proteins or protein channels. Otherwise, you might be embedded or you could just be on the surface. So this represents a protein on the surface, and it could be on the inside or the outside of the cell's membrane. And what might this circular thing be? Some other small molecule that might be interacting with a protein to cause some chain of events. So lipids in their truest sense are not going to be very functional without specialization. And the specialization comes from the proteins. What we're trying to do with this whole section is understand how life works. So the last two sections are really what most people would consider life molecules. So if we go with, let's name some proteins. Is DNA a protein? No. Is RNA a protein? No. Is hemoglobin a protein? Would you say it's important to life? Why? Because it's an oxygen carrier in the blood. So proteins have functions. They sort of get named historically or by their function. So if we say we have structural proteins, would you say your fingernails are important? How about your hair? How about collagen? So ligaments, would we say are structural? Hair, structural. Fingernails, structural. In that sense, would we like them to dissolve? But hemoglobin, not structural, is what we would call a carrier protein. So since it's in the blood, as Tia Rose said, do we like it to dissolve? And do we like the oxygen to be carried, or do we like the oxygen to actually react with the hemoglobin? So the key thing about hemoglobin is oxygen can bind when oxygen pressure is high. That would be in your lungs, but can the oxygen equally as easily fall off when the oxygen pressure is low in your cells? So in that sense, is oxygen binding reversible in hemoglobin? So we don't really want the hemoglobin to react permanently with the oxygen. We want it to be able to bind release. So our proteins kind of tailor-made to suit their function. Can we also eat proteins for energy? Do we need proteins for our immune system? So in the news lately has been COVID. And would we say the spike protein in COVID is a protein? The name kind of gives it away, spike protein. What does the spike protein interact with in our bodies? Does it have to bind to certain cells for the coronavirus to get in the cell? If it doesn't bind well, would you have quote unquote natural immunity? If it binds well, but you have immunity, would your immune system create a cytokine storm to try to remove the coronavirus? So are proteins involved in immunochemistry? So your receptors are proteins, but they get special names like antigens and antibodies. That's the response if an antigen is acted on. So, well, you won't get the disease, or you'll get a milder form of the disease. So, in other words, if the spike protein couldn't bind to your cells, then you don't really need immunity because the spike protein can't bind and the virus can't get in. If you've been vaccinated or you've previously had COVID, then hopefully you have some immunity, meaning if the spike protein binds, your body's immune system remembers it. As soon as it gets bound, the bells go off and your immune system goes after it and takes care of it. If you don't have immunity, then your immune system doesn't recognize it and lets it do its thing. So of course I missed some. Do we have proteins like actin and myosin involved in muscle and movement? And we covered transport, storage, protection, regulatory, and the big one I want to focus on are enzymes. So basically, an enzyme can be a protein that catalyzes some enzymes are RNAs and DNAs, but certainly all proteins that catalyze reactions are classified as enzymes. And when there's many classes of enzymes, but almost all proteins that are enzymes end in ACE, ASE. So if I say sucrose is what? Sucrose is a disaccharide. And if you eat sucrose, guess what enzyme will act on it? Sucrase. So sucrase would be an enzyme, which is a catalytic protein that catalyzes the hydrolysis of sucrose. So how about lactose? What enzyme would act on it? Lactase. So if you're lactose intolerant, what enzyme don't you have or don't have a lot of? You don't have a lot of lactase. So are there a multitude of proteins which are enzymes? One important one for our respiratory system would be carbonic anhydrase. So what would you guess it acts on? Carbonic acid. So carbonic anhydrase catalyzes the decomposition of carbonic acid into CO2 and water, and that allows us to breathe out CO2 at an appreciable rate. If we couldn't get rid of CO2, what would happen? Well, life as we know it couldn't exist, because why do we need to breathe? To live. What's more important, getting oxygen in or getting CO2 out? CO2 out. So if the CO2 in your body is trapped as carbonic acid, is it harder to get it out because carbonic acid's not a gas? So does the enzyme carbonic anhydrase speed up that reaction? So without enzymes, which again are catalytic proteins, could life as we know it exist? Not as we know it. And are we interested in the structural components of proteins? We are. So proteins are polymers of what? Amino acids, and as the name suggests, two amino acids have an amine group and an acid. And there's 20 amino acids, and by convention, always draw your amino group on the left and your carboxylic acid on the right, and have them separated by a C, an H, and an R. And of course, with that said, are most of our amino acids chiral? They are, and for whatever reason, way back when, nature chose L. So your naturally occurring amino acids are all L. Since they make proteins, are your proteins chiral? They are, and we would say they're all derived from the L enantiomer. And is L R or S? Usually S, but again, with sugars and macromolecules, we fall to the DL system. What you do have to know is amino acids are chiral, but nature chose the L. And importantly, if we look at an amino acid, the simplest one has these two groups on the C as H's, and that's glycine. So if we look down here, the amino acid called glycine, sometimes we want to use abbreviations for when we get to nucleic acids, sometimes 3 letters, sometimes 1 letter. And there again, the structure is. Other amino acids don't have 2 H's, but rather have an R group replacing 1 or more of the H's. So as we go through these, alanine has this methyl group, valine has this isopropyl group, leucine has this sec-butyl group, isoleucine has isobutyl, proline is one of the unique ones in that it has this wrapped around version, which wraps around back to the amine. So proline is a cyclic amino acid. Phenylalanine has this phenyl connected to the carbon, This whole group we would call benzyl, but it's called phenylalanine because if you go back and look at alanine, it's a methyl group. So if you connected a phenyl group to that methyl, that's how the name phenylalanine was derived. Phenyl group on the carbon, which was methyl, becomes phenylalanine. And we have methionine, which is a sulfur ether. And then certain amino acids have what are called polar groups on the side. Classic polar group is OH. So we have serine, threonine, cysteine is a thiol. That's really important. Tyrosine, asparagine represents a group which has on the side chain an amide. Glutamine is an amide, and tryptophan has this indole group on it. And then we have certain amino acids which are quote unquote acidic, meaning in addition to the carboxylic acid of the amino acid is this carboxylic acid on the side chain. And we have glutamic and aspartic acid. And then certain amino acids are basic: lysine, arginine, and histidine. So there are your 20 amino acids. You certainly don't have to memorize all their structures, but you should familiarize yourself with the names and or the general formula. And the reason we're studying amino acids in depth is because they are linked with amide bonds to make what are called peptides, polypeptides, and proteins. And again, proteins are the molecules of life. So to understand protein chemistry means we need to understand amino acid chemistry. One thing about amino acids is these would be neutral forms. This is truly neutral in the sense that there's no charges anywhere. But as we saw last week, this amino group has basic tendencies and this carboxylic acid has acidic tendencies. So you can get an internal proton transfer to make this charged, doubly charged molecule or form. And this is called a zwitterion. You can debate a lot, but it's just important to be able to recognize that these both exist. And they can exist simultaneously and equilibrate with each other. When we go to link two amino acids together, this is why the convention is important to follow. Always draw the amino group, then the carbon and whatever specific amino acid it is, then the carboxylic acid of your first amino acid, and then your second amino acid, draw it the same way, because what you're setting yourself up to do right now is connect this nitrogen to this carbon using an amide bond. So if we stop right there and say, this is a dipeptide because we've connected two amino acids, what molecular functionality have we used to connect them? An amide bond. So proteins are nothing more than polymers of amino acids. And what bonding pattern connects the amino acids? Amides. And why would we call this a dipeptide? Because right here we have one amino acid. Here we have another amino acid. Before they're connected, are they just two amino acids? Once they're connected, it's a dipeptide because we've connected two amino acids. So if that was fun, we do it again. In this, how many amino acids are now connected? 3. How many amide groups are there? 2. And would we call this a tripeptide? And now for the details. This R group dictates the N terminal because it has a free amino group. This R group would dictate what we call the C terminal because it has a free acid group. This R group is the middle of the two, so it's neither N terminal or C terminal. So now if I put some specifics together, if I look at this tripeptide, this is the R group. Which is the first amino acid in the chain. This is the R group of the second. This is the R group of the third. So this R group, which is derived from aspartic acid, is the N terminal. This R group, which is derived from glycine, is the C terminal. And this R group, which is phenylalanine, is neither N terminal or C terminal. It's in the middle. So we would call this tripeptide asp-phe-gly. So flipping the script, if I say asp-phe-gly, which is the N terminal? Asp, which is the C terminal? Gly, because we're always listing from N terminal towards C terminal, which is why, once again, is it very important when you draw your amino acid to start with the free amine group, then a carbon with an R group, then an acid. And then when you draw your second amino acid, what goes on the left? Amine group, then carbon, then acid. Thus, are we connecting the acid of the first with the amine of the second? And when we say we're connecting, what kind of connection are we making? Amide bond. So is asp-phe-gly different than phe-gly-asp? They would be totally different tripeptides, even though they had the same amino acids in them. So said another way, is the order of amino acids critically important? Yes. Of course, does a protein have way more amino acids than just 3? Sometimes are there thousands. So is it random how they're connected, or is it orchestrated? It's going to be orchestrated by the next class of macromolecules, which are your nucleic acids. But before we get there, we just want to summarize protein structure. So proteins are said to have 4 types of structures. The primary structure is defined as the amino acid sequence. The secondary structure is repeating patterns caused by looping around an intermolecular or intramolecular amide or hydrogen bonding. The tertiary structure is the overall 3-dimensional shape, and the quaternary structure is how oftentimes one or more protein subunits can interact with each other. So if we've picked up nothing else so far, are the shapes important? So is the tertiary structure, aka the overall 3-dimensional shape, really important for the function of a protein? So to summarize, protein chemistry is function all about shape. And is shape dictated by structure. So we must know a protein's amino acid sequence because that structure ultimately dictate the overall shape of the three-dimensional protein. And why is the shape critical? Because then it's all about if things are shaped to fit, will molecules interact with proteins and make things happen? And that's what life's about, especially with our catalysis and our antigens and antibodies. And who's ever cooked an egg? So if you have an egg, what color is it? Clear and a yolk. And so why do we call it an egg white? When you cook it, it turns white. And so have you done a chemical reaction? Possibly. Have you certainly done a denaturation? So when proteins are in their native state, natural, we call them native. If you do anything to them to take them out of their native state, you have said to denature the protein. So is heat one way to denature a protein? So when you cook your egg white, which starts off clear, would you say the protein in the egg white is native? When you cook it, are you denaturing it? Can you uncook it and renature it? Not easily, but are some proteins able to be renatured? Yes. So things that have an important function, is it important to keep them in their native state? If you denature your protein, a lot of times do you lose the function? Confusingly, you may not have altered the actual amino acid sequence. So you may not have done a chemical reaction. You may have just tinkered with the overall three-dimensional shape. So denaturation doesn't have to imply you've done a reaction. You've just somehow altered the protein structure and or shape. And why don't we like our proteins to be denatured in our body? Because proteins are the molecules of life. So if we want our proteins to help us live, we want them in their native state. So what else can denature proteins? Light, heavy metals, etc. And so on Wednesday, we're going to go through nucleic acids, which is a summary term for RNA and DNA, which gets back to, are proteins synthesized randomly or orchestrated? So who orchestrates the protein synthesis? The DNA and RNA. And then what ultimately enzymatically is involved in controlling the DNA and RNA and synthesizing proteins? Other proteins. So then the main $64,000 question is, which came first, the protein which controls the DNA or the DNA and RNA which makes the protein which controls it? And is there a correct answer for that, or does nobody truly know? And can you then make it a nice essay question? So on Wednesday, we're going to go through the basics of RNA and DNA and their structural entities. And then with whatever time, we'll start working on the problem set.
So,
And everyone has the macromolecule handout,
so
so you can certainly read
going
ahead.
to be included in the test. Hello.
What? I'm going to kill the projector, then I think we'll be able to hear Valeria's question.
Okay.
Right now, it's trying to use this.
So is all this going to be in the test? Hi, can you hear me?
Hey, Valeria, I think we heard you say something, but we couldn't hear exactly what you said. Can you hear me?
Yeah, can you hear me? Can you hear me? Can you hear me?
I can now. I'm sorry.
That's okay.
Can you?
So all this that we did today, is that going to be included in test number 4?
Yeah.
And
Yeah. If you,
I
I sent everyone an
have
email with the
other
practice problem.
questions.
Well, now you're
Okay,
breaking
yeah,
up,
I
so I
printed
didn't
out
hear
last
anything.
night. I haven't seen it yet. So are we having a lab next Monday too during finals? Sorry, my internet is really bad.
Yeah, we agree
Are
with
we
that.
having a lab next Monday too, or today's the last lab? Because I'm not clear
Next Monday is going to be
from
open
quarantine
lab.
until Wednesday. That's my last day. Because it'll be 10 days from my loss of sense of smell. Cool.
Hallelujah.
And then are we still meeting Monday morning?
Yes. Yeah,
Okay.
Monday
And
we'll
the
get back
test,
to score and go and review for the final, and then Monday afternoon next week we can do whatever, review, do lab, whatever you want to do.
okay. And then test number 4 is this Friday, correct?
Yes.
Okay. I just wanted to review those things.
Okay, send me an email if you got more questions, because I'm going to kill the session.
Okay, thank you. Have a good day.
You too. Glad you're feeling better.
Yeah, me too. I mean,
Okay.
it's still a little bit of shortness of breath.